Right, moving on to mitosis. Now as we did, this cell is now ready for division. So I've just drawn this cell over here, the cell after interphase, ready for division, has doubled up its cytoplasm and has doubled up its DNA. Okay? It's going through mitosis now, which makes two exact copies of this cell. This cell is called the parent cell, and the two that come out of it are called the daughter cells. So it's going to make two daughter cells. Again, mitosis is also an elaborate process and is further divided into 4 stages. The first one is prophase. Yeah, prophase. So this cell is going to go through prophase. Lots of things happen in prophase. The first one is chromosomes condense. Condense means they start losing water and they start becoming more visible. Okay? So as I've shown them over here, they're not actually like this at this stage. So as they go towards mitosis, prophase, that's how they're going to look like. Thicker and shorter. More visible by losing water, and that's called condensation. Right? The other thing that happens is this nuclear membrane starts to break down. So nuclear membrane breaks. Ultimately, this whole thing is going to go away, and the chromosomes will be sitting in the cytoplasm. Okay? The third thing that happens is two organelles called centrioles. They start moving apart, so one goes to this side and the other one to the other side, and they develop a spindle in between. So spindle is formed. Spindle is formed. And the fourth thing that happens is these chromosomes attach themselves to the spindle. Chromosomes attach to spindle. Do you know what part of a chromosome attaches to the spindle? Centromere. Good. So this thing, this part, is called the centromere. This will make the cell look like this. So we have the cell membrane, nuclear membrane is gone, centrioles go to the opposite ends, they develop a spindle which is made up of special protein, and the chromosomes attach themselves to the spindle. Yep. Next stage. What's the next stage? Metaphase. Only one thing happens in metaphase of mitosis, and that is all of them line up in the middle. So that would be just drawing the same cell. Mid. Yeah, they come to the middle. Yeah. So they line up in the middle, and the middle is called the equator. Right? What's the next stage? Anaphase.
And you know A, that was for apart, so me and then apart.
Yeah, oh good. A for apart. Two things happen in anaphase. Number one, these two chromatids, what are they called? They belong to the same chromosome? Sister chromatids. So these two are called sister chromatids. The two that belong to the same chromosomes. Okay? The sisters separate. And the other thing is they move to the opposite ends. They move apart. So sister chromatids separate and move So how do we draw it? There, got the spindle. Split each of the chromosome into 2 chromatids. And then take one chromatid to this side and the other one to this side. So if you are asked what is the stage when the centromere splits, the answer is anaphase. Okay? Because it's the splitting of the centromere that separates the sisters. Right? So this goes here, here, and the other. The last one is telophase. Telophase is kind of like the opposite of prophase. So in prophase the spindle was formed. Now the spindle has done its job. And if you're asked what's the job of the spindle, why was spindle formed?
So chromosomes should
To help pull them
Separate equally. Okay? So that they could be distributed equally between the 2 doors. Right? See the spindle, it attaches the chromosomes, and then once the, say, one chromosome was here, the spindle fibers from here and here, they start contracting, and that pulls the sisters apart. Right? So the spindle organizes the breakup of the sister chromatids equally, and they're going to different ends. So this spindle fiber would have contracted, this one would have contracted, and that's how they pull the sisters apart. Right? So in telophase, the spindle goes away because it's done its job. The nuclear membrane starts being formed around the 2 sets of chromosomes. We've got 2 sets of chromosomes now, and everything is going to be reversed. So nuclear membrane comes back. The spindle goes away, chromosomes start absorbing water, and they again become thread-like, and yeah, this one's done. That's telophase. Along with telophase, so that's about it. That's about mitosis. And then along with that happens another process called cytokinesis, which is like cutting off of the cells to make tea. Right? So this one is finally going to end. The final stage of telophase, along with cytokinesis, would be this, that, and then new centrioles come up and all that cytoplasm stuff. Yes, ma'am.
Do you know how a plant though has a ribbon wall?
Yeah.
How does it, because you know how this one has a self-borrowing integration, how does it happen in a plant though?
Good question. That's exactly what I was about to explain now. The other question that you could be asked is, how is the division in a plant cell different from the division in an animal cell? So, because the plant cells have an additional wall around them, I'll rub this off. Up to the telophase stage, everything is the same. Even telophase is the same. Cytokinesis is different in a plant cell. Okay? Because the plant cell is like this, for example, and it has an additional hard, rigid wall around it. So you cannot contract the plant cell from here. You can't shrink it. Okay? So it doesn't, cytokinesis does not happen in the same way in a plant and an animal cell. So what happens instead is, once this thing is happening, the 2 nuclei, daughter nuclei are being formed. The plant cell makes an entirely new wall in the middle. Okay? And that's organized by the organelle, which is Golgi apparatus. So Golgi apparatus, it collects a few tubules here and there, and that makes, say, a new cell wall starts coming up here. With the membrane. And then finally it gets cut off. Okay? So that's going to be.
So you said the Golgi apparatus makes it into a wall.
Yes, it helps make it. new cell wall. And it's going to be cut from. So that's the difference in cytokinesis between the plant cell and the animal cell. Yeah? So this finally, in both the cases, results in two cells which are the exact copies of the original parent cell. We started off with a parent cell that has how many chromosomes? 4. We end up with two daughter cells having how many chromosomes? 4 each. Okay? So it's an equational division because it results in exact copies. That is mitosis. Any questions?